Have you ever wondered where genes come from and how they are created? It's a question that's been captivating scientists and philosophers alike for centuries. Genes, those microscopic of DNA, are the master architects of every living organism. They are the blueprint, the instructions that guide the construction of everything from the color of your eyes to your predisposition to certain diseases. For example, it's your genes that decide whether you eyes or brown, blonde hair or black, or whether you're tall or short. These traits, among countless others, are determined by the subtle variations in our genetic code. They are the unseen threads that weave the tapestry of life, dictating our traits and characteristics, influencing our health, and even shaping our behavior. But what exactly are these genes? Picture them as recipes in the grand cookbook of life, each one outlining specific instructions on how to build a protein, the building blocks of our bodies. There are estimated to be around 20,000 to 25,000 of these recipes in the human body. Each one unique, each one vital. Now, understanding genes and their importance is one thing, but comprehending the complexity of gene creation and manipulation is quite another. It's akin to trying to understand the intricate workings of a grand orchestra, where each musician plays a crucial role, but it's the collective symphony that creates the magic. In the way, genes don't work in isolation. They interact with each other and with the environment, creating a dynamic landscape that is far from being fully understood. The mere creation of a single gene is a marvel of biological engineering, a process that involves numerous steps and a host of different molecules. And what about the idea of changing our genes to suit our needs or desires, whether it's to cure diseases or adapt to changing environments, may sound like science fiction, but with advancements in genetic engineering becoming increasingly possible. But how exactly are these genes created and can we manipulate them? Let's dive into the fascinating world of genetics. We are MindArt100. Subscribe to us and allow us to send future notifications. Genes don't just appear out of nowhere, they have a specific origin and a complex process of creation. Let's illustrate this journey with a real-world example. The creation of insulin, a critical hormone that regulates blood sugar levels. The birth of the insulin gene is a captivating venture that starts with DNA replication. The DNA, our genetic blueprint, unwinds its iconic double helix structure. Here, an enzyme called DNA polymerase reads the DNA strands and creates new copies. This process is like a meticulous librarian, copying word by word, or in this case, base by base, the instructions for making insulin. The four bases, adenine, thymine, cytosine, and guanine, pair up in a specific sequence, creating a mirror image of the original insulin DNA strand. Post this, we have two identical copies of DNA ready to pass on the instructions. However, the birth of the insulin gene doesn't stop at replication. The DNA then undergoes a process called transcription, which is like translating the language of DNA into a slightly different dialect, known as RNA. For our insulin gene, the enzyme RNA polymerase creates a single-stranded RNA copy of the DNA, which is like a messenger, carrying the instructions from the nucleus to the rest of the cell. Here, in a process called translation, the RNA message for insulin is read and used to assemble amino acids into the insulin protein. Each step is vital, each enzyme a key player. A single error can lead to malfunctioning insulin proteins, potentially leading to diabetes. But when everything goes right, the result is the insulin gene, a specific segment of DNA that codes for insulin. This gene is one of the countless architects of our bodies, the writers of our biological story. They determine not just our hair color and height, but also how our bodies function on a cellular level. They are the essence of life's diversity, creating countless variations from the same basic blueprint. This intricate process results in the creation of unique genes, like those for insulin, that make us who we are. Now that we understand how genes are created, can we alter them to our liking? This question brings us to the captivating realm of gene manipulation. Imagine being able to rewrite our genetic code, akin to a software engineer refining a complex program. A thought that has captivated scientists for decades and has now turned into reality. Gene manipulation, or genetic engineering, is a process that involves modifying an organism's DNA to bring about desired traits. For instance, using this technology, crops can be made more resistant to pests, paving the way for a revolution in agriculture. This is not a hypothetical situation. Scientists have utilized CRISPR, an innovative tool short for clustered regularly interspaced, short palindromic repeats, to create disease-resistant crop varieties. Borrowed from bacteria, this molecular machinery allows us to cut and paste DNA with precision. It's like a pair of molecular scissors that enables us to add, remove, or alter specific parts of the genetic code. The implications of this technology are enormous. Diseases that were once thought incurable could potentially be eradicated. We could engineer plants to withstand harsh environmental conditions or create bacteria that can decompose pollutants. The possibilities seem boundless, but with great power comes great responsibility. The ability to manipulate genes raises a series of ethical questions. If we can edit genes, should we? And if so, where do we draw the line? These are questions that society as a whole must grapple with. Moreover, there's a lot we still don't understand about the complex interplay of genes. Making changes to one part of the genome could trigger unforeseen consequences elsewhere. It's a bit like playing a high-stakes game of dominoes. In conclusion, gene manipulation is a double-edged sword. It promises immense potential for good, but also carries significant. As we continue to explore this brave new world of genetic engineering, we must tread carefully, balancing our ambition with caution. Manipulating genes isn't just a concept from science fiction. It's a reality that brings immense possibilities and challenges. Our genes aren't just the product of a biological process, they're also influenced by our environment. But how? Let's start by understanding the concept of gene-environment interaction. In a nutshell, it refers to the way in which our genes and external factors play off each other. Picture it like a dance. Our genes aren't dancing solo, they're dancing with the environment in a complex, elegant waltz. External factors can influence gene activity and expression in a variety of ways. For instance, exposure to certain chemicals can turn genes on or off, affecting how they function. This is a process called epigenetics, where changes in gene expression occur without alterations in the DNA sequence itself. Let's take diet and exercise, for instance. Studies have shown that a diet rich in fruits, vegetables, and whole grains, along with regular physical activity, can influence the expression of genes associated with disease risk. For example, these lifestyle factors may downregulate genes associated with inflammation, thereby reducing the risk of chronic diseases like heart disease and type 2 diabetes. Conversely, a diet high in processed foods and sedentary behavior can upregulate these same genes, potentially increasing disease risk. Diet, stress, smoking, alcohol, exposure to sunlight, all these factors can leave epigenetic marks on our DNA that modify its activity. It's fascinating to think that the choices we make, the food we eat, the air we breathe, can all leave a lasting impact on our genetic code. It's like our genes have a volume control. External factors can turn the volume up, enhancing gene activity, or turn it down, dampening it. On the other hand, chronic stress can ramp up activity of genes linked to inflammation, potentially increasing the risk of conditions like heart disease and diabetes. But gene-environment interactions aren't just about the negatives, they also offer opportunities for positive change. If we can understand how these interactions work, we could potentially leverage them to improve our health and well-being. We could adjust our lifestyle to optimize our genes, or even develop therapies that can modify gene activity in beneficial ways. It's an exciting field of research, one that holds the promise of personalized medicine, tailored to our unique genetic makeup and lifestyle. And remember, our genes are constantly interacting with our environment, creating a dynamic and ever-changing genetic landscape. With advancements in science and technology, what does the future hold for gene creation and manipulation? In the light of scientific discoveries, the realm of genetics is on the brink of breakthroughs that once seemed the stuff of science fiction. As we peer into the looking glass of the future, we see the potential for unprecedented developments in gene creation and manipulation. Imagine a world where we could engineer genes to adapt to environmental changes. Climate change, for instance, is a pressing issue that threatens biodiversity. In the future, we could potentially create genes that help organisms adapt to rising temperatures or to new diseases that may emerge as a result of changing ecosystems. This could be a game changer in preserving our planet's rich tapestry of life. Moreover, advancements in gene manipulation might usher in an era of personalized medicine. We could potentially tailor treatments to an individual's genetic makeup, enhancing their effectiveness while minimizing side effects. This could revolutionize how we approach diseases, from cancer to rare genetic disorders. An example of this is how gene therapy is currently being used to treat genetic disorders like cystic fibrosis. By introducing healthy genes into the patient's cells, This groundbreaking treatment can significantly improve the quality of life for patients, giving hope to those living with this previously untreatable condition. But it's not all about the physical. Consider the implications for mental health. We could potentially manipulate genes to understand and perhaps even prevent mental health disorders. This could change our approach to mental health, moving us closer to a world where no one has to suffer silently. However, as we stand on the precipice of this brave new world, it's crucial to remember the ethical considerations. Manipulating life at such a fundamental level comes with great responsibility. We must ensure that these technologies are used for the benefit of all, not just a privileged few. We must also consider the potential for unintended consequences. After all, the genes we create and manipulate don't exist in isolation. They interact with each other and the environment in complex ways. The future of genetics holds immense promise, but it's a future we must approach with caution and responsibility. As we continue to unlock the secrets of our genes, we must also ensure that we wield this powerful tool with the utmost respect for the intricate dance of life. Genes are more than just biological blueprints. They're powerful tools that can shape our lives in countless ways. They're the unseen architects, silently pulling the strings behind the scenes of our lives. From the color of our eyes to our susceptibility to certain diseases like cancer and heart disease, genes play a critical role in defining who we are. Imagine genes as a set of instructions, a recipe that dictates our physical and, to some extent, our psychological makeup. They influence our growth and development, our behaviors, and even our personalities. Every trait we possess, from our height to our temperament, is influenced by the complex interplay of the thousands of genes that reside within our cells. In the realm of health, genes hold immense power. For instance, they can make us resilient against certain diseases, yet susceptible to others like cancer and heart disease. They can also determine our body's responses to medicines, influencing how effectively we can combat illnesses. On a broader scale, understanding the genetic underpinnings of diseases like cancer and heart disease can lead to revolutionary treatments and even preventative strategies that can nip these diseases in the bud before they manifest. But it's not just about health and disease. Genes can also influence our behaviors and personalities. Have you ever wondered why some people are more outgoing while others are more reserved? Or why some people seem naturally inclined towards music, art, or mathematics? The answers lie, at least in part, within our genes. The potential of genetic research is vast. As we continue to unravel the mysteries of the human genome, we open doors to a future where we can better predict, prevent, and treat diseases like cancer and heart disease. A future where we can understand ourselves and others on a deeper, more profound level. A future where we can harness the power of our genes to improve human health and well-being. The power of genes is immense, and understanding them can unlock new possibilities for our future. We've journeyed through the intricate world of genes, from their creation to their manipulation. But what does all this mean for us? In the grand tapestry of life, genes are the threads that weave together the story of who we are, where we come from, and what we might become. They are the architects of life, the silent puppet masters pulling the strings behind the scenes of every living organism. We've traced the birth of genes, from the primordial soup of life's earliest days to the complex beings we are today. We've seen how genes are not just static blueprints, but dynamic entities, changing and adapting over time. They are not merely the product of evolution, but the drivers of it, shaping life in response to the environment and the needs of the organism. The art of gene manipulation, a feat once thought to be the stuff of science fiction, is now a reality. We've dabbled in the very essence of life, tweaking and modifying genes to serve our purposes. From creating drought-resistant crops to curing genetic diseases, the possibilities are as vast as they are exciting. For instance, understanding our genetic makeup allows for personalized medicine, treating patients based on their unique genetic profile. This can predict disease risk and manage treatment effectively. But with great power comes great responsibility. The potential of gene manipulation is immense, but so too are the ethical considerations and potential pitfalls. We've seen how genes interact with the environment, how they're not just passive recipients of environmental cues, but active participants, changing and adapting in response to their surroundings. This is a dance as old as life itself. a delicate balance between stability and change, survival and extinction. Looking to the future, we've pondered the possibilities and challenges of gene creation and manipulation. The potential to shape life to our liking, to create new forms of life, to cure diseases and prolong life is tantalizing. But the path is fraught with ethical and practical challenges. How do we navigate this brave new world? How do we ensure that we use this power responsibly for the benefit of all life? The power of genes is immense. They shape every aspect of our lives, from our physical traits to our susceptibility to diseases, from our behavior to our lifespan. Understanding and harnessing this power is one of the greatest challenges and opportunities of our time. We've journeyed through the fascinating world of genes, delved into their birth, their manipulation, and their interaction with the environment. We've glimpsed the future, a future shaped by our understanding and manipulation of genes. But we're just scratching the surface. The world of genes is as vast and complex as the universe itself, a universe waiting to be explored. The world of genes is as complex as it is fascinating, and it's a world that we're just beginning to understand. We stand on the brink of a new era, an era shaped by our growing understanding of genes. The journey ahead is filled with excitement and challenges, and we can't wait to see where it takes us.